Welcome everybody to today. I'm super excited to be able to introduce this panel. We have Michael Funk from Science. He's a senior editor, a very well-seasoned senior editor at Science, has been working in the science area for a long time. On our panel today, we're talking about protein degraders. Protein degraders, we think, has been born here at Yale. Unfortunately, we don't have Craig Cruz here. We'd love to have him, but he's flying around the world. But we have the people that are advancing these protein degraders as drugs, which is really, really important for us to hear that there is an endpoint for all the wonderful research that's going on here at Yale. I'm going to hand over to Michael and let him moderate, and have fun, guys.
Great. Thank you so much. Hello, everyone. My name is Michael Funk. I'm from Science Magazine. I'm a senior editor there, and I handle sort of basic science research in biochemistry. So this has been an exciting meeting for me, getting to hear from people doing sort of things in the more translational space and investors interested in these technologies. So I would just like to start off by having everyone introduce themselves, and then we'll get into some questions. So Adam?
And slides, too.
And, yes,
right?
and slides. Yeah.
Yeah, hi, I'm Adam Gilbert. I'm an external science lead for Pfizer. My job is really looking at the external academic and biopharma landscape and trying to come up with the best platforms to align with the therapeutic strategy and targets that Pfizer wants to prosecute. I do this for assets and as well as platforms, as I just said. I've been doing that for the past 3 to 5 years. Before I did that, I led a platform medchem group within Pfizer. TPD was one of the main things we focused on. I've been involved in the collaboration that Arvinas and Pfizer have had for quite a long time. I'm also big into covalent inhibitors if we want to have a tangent session besides this one. But anyway, it's a pleasure to be here.
Wonderful, thank you. I think next was Angela.
Angela Cacace. I am the Chief Scientific Officer at Arvinas. I've been there for 7 years, really spearheading the neuroscience focus at Arvinas, but really proud of all the work that we've accomplished as a degrader company spun out of Craig Cruz's lab. So very excited about the progress that we've made in the targeted protein degrader space. In fact, tomorrow the pivotal study, the phase 3 study, is going to be presented for the first proximity-induced degrader, vepdegestrant. So it's an exciting time. And we're benefiting patients, so that's what really matters. Prior to joining Arvinas, I was at a small biotech for 2.5 years called Therapeutics, where we focused on epigenetic regulation in rare disease. And then prior to that, 20 years at Bristol Myers Squibb in various capacities, but working mostly in drug discovery, focused on neuroscience and also rare disease. So, and then a year sabbatical at Pfizer, that's what I call it, my year sabbatical at Pfizer, working on an antibody project in oncology.
Everybody works at Pfizer at some point in their career.
Yes, yes, and I trained at Columbia University, and my PhD is in pharmacology. That's me.
Okay. And I think Danette.
Yeah. Hi, I'm Danette Daniels. I am a vice president of the protein degrader platform at Foghorn Therapeutics, which is located in Cambridge, Massachusetts. I lead the degraders that are headed to clinic for a variety of oncology indications at Foghorn Therapeutics. I also lead a broader initiative within induced proximity. I am classically trained as a crystallographer. I got my PhD here at Yale in the molecular biophysics and biochemistry department. I went on into crystallography for quite a while, but then transitioned to a job at Promega Corporation, where I did a lot of technology development. I got started very early on in targeted protein degradation. Actually, with Craig was the first collaboration on what many don't know, but were hydrophobic ligands at the time, so almost 15 years ago, in attempts to try to degrade proteins. We've subsequently moved away from that to the E3 ligases, but it's been really a great time to be in targeted protein degradation, to kind of witness it from that very initial start. to where it is now. And I'm really excited about the opportunities to take degraders to clinic and develop them, because I think there's a lot of power to benefit patients.
Wonderful, thank you. Vlad?
Hello, everyone. I'm Vlad Coric. I'm the CEO of Biohaven. I'm a physician by training, did my residency and fellowship at Yale, and then ran a clinical research unit at Yale for a number of years before going to Bristol Myers Squibb, where I got to work with Angela. Didn't work at Pfizer, but sold the drug to Pfizer, so that's not bad.
I haven't done anything.
No.
Oh, I collaborated with Pfizer in my early days.
We went out to dinner last week.
Oh, yeah, we went.
And it was really within this Yale ecosystem that I first heard about degraders. And Angela's the other bookend of the intracellular, which years back, no one was interested in extracellular degraders. Everyone and all the talk was intracellular, and I got to watch all of Angela and Craig Cruz's great work. And in the background, we were advancing these extracellular degraders that you'll hear about today, and I'm particularly excited about it because of the impact it will have on patients. And we presented some new data that we'll show you a hint of here today of our first human data demonstrating how we're going to leverage this to bring precision immunology to patients.
Wonderful, thank you. So I think we had some slides. I don't know if we want to do that
some
now.
Slides.
I think the
clicker.
advancer is right there.
Okay. Let's see. Okay. So I wanted to take the opportunity to bring back to the Yale community some of the data that we've been generating for our targeted protein degrader platform, the proteolysis targeting chimera. And this allows us to selectively ubiquitinate proteins and remove them from cells. And so we're going after disease-causing proteins in oncology and neurology. And so today I'm going to tell you a bit about the pioneering work that we've been doing with the first PROTAC that's actually in neurodegeneration right now. So I'll show you some of the data to selectively clear a pathologic protein from the brain. So oral ARV-102 is the first brain-penetrant investigational PROTAC degrader of LRRK2. LRRK2 is genetically implicated in both Parkinson's disease, so it's at the root cause of late-onset Parkinson's disease, and also progressive supranuclear palsy, which is a devastating tauopathy that generally causes death within 5 to 7 years post-diagnosis. And it's just a continuous decline in function over time. And so if you could develop a PROTAC that targets a protein that removes the pathologic protein, in this case LRRK2, you could potentially take the brakes off the lysosomal clearance system and remove pathologic proteins. You could also potentially impact neuroinflammation, which is also a very key component of neurodegeneration. So we've been able to show in non-human primates that our orally bioavailable PROTAC degrades LRRK2 across the primate brain, including deep regions of the brain where genomic technologies struggle to get. And so, of course, we're orally administered. We don't require intrathecal dosing like antisense oligonucleotides. So it's an exciting time in targeted protein degradation, particularly with the accomplishments of the team, the innovative team at Arvinas that really engineered these PROTACs to cross the blood-brain barrier. And so what we've been able to show with our PROTACs is that we do induce lysosomal degradation, and we do so in a way that is differential from inhibitors. We also induce lysosomal number, and we've been able to show that we reduce LRRK2 in cerebrospinal fluid, which is the collectible fluid. So you can see after a single dose in non-human primates that we reduce LRRK2 in 2 out of 3 animals, and then over 14 days daily dosing, we get down to baseline. And we've also been able to identify pathway biomarkers that we think are really important. One of the most important things in translational neurodegeneration is to really ensure that you're able to modify or modulate pathways that are involved in disease. And we've been able to identify pathway biomarkers that are linked to LRRK2 in the Michael J. Fox Progressive Marker Initiative from cerebrospinal fluid from patients, and then take those markers and say, do they change in CSF from non-human primates with elevated LRRK2? And I'm showing you IBA1, which is an important inflammatory marker that surrounds every major neurodegenerative protein in every major disease. So it's exciting times. We've been able to also show that we impact markers of leaky lysosomes as well as neuroinflammation. I mentioned IBA1, GPNMB, which is another marker. And in an exciting manner, we're also seeing elevation of markers that are involved in energetics as well as synaptic markers. So these are the human data. This is our phase 1 healthy volunteer data. We've finished our phase 1 study. And what I'm not showing you is that we achieved dose-dependent increases in ARV 102 in CSF. And what we've observed in our single and multiple ascending doses is that we dose-dependently reduce LRRK2 in CSF, even after a single dose at reasonably low doses. So right in the SAD, greater than 60 milligrams in the multiple dose, 20 milligrams oral dosing gets across the blood-brain barrier in a dose-dependent manner and reduces the target at our target, which is 50%. So we have a lot of leeway with our small molecule to really optimize dosing. And it's really exciting to see these data in healthy volunteers. We're now in Parkinson's disease patients. So it's an exciting time at Arvinas in neurodegeneration. We've also been able to move pathway markers. I'm not going to take too much more time, but just to suffice to say that the incredible research team at Arvinas is constantly looking to move and pioneer our PROTAC modality in cancer, in high unmet need, and also in neurodegeneration. So it's an exciting time at Arvinas. So thank you very much for that opportunity to present our data.
Yeah, thanks. Great to see some data. I think Danette is next.
Yeah, so I just wanted to give a brief overview of how Foghorn Therapeutics is thinking about employing targeted protein degradation. We use a lot of modalities at Foghorn. We are focused on the chromatin regulatory system. And if you think about the action of drugs, ultimately there's some type of transcriptional readout that occurs that is really controlling the disease. So we thought, go to the heart of it. We'll go after these chromatin modulatory proteins. So we are targeting proteins within chromatin remodeling complexes, such as BAF, or other modulators which deposit histone marks or to either activate or repress chromatin. Or, as you will see, a space that really degradation has been very successful, especially with molecular glues, is going after transcription factors. So one of, a theme that is in our space really is using degradation to achieve selectivity, especially when selectivity is very difficult with inhibitors. And that's because a lot of the proteins that we're going after have very close related homologs. The domains that you would target with binders are not good at opening the therapeutic window between targeting one and not the other. So we have just 4 programs listed here: SMARCA2, ARID1B, CBP, and EP300, and we all have selective degrader programs for each of these. Our SMARCA2 program is partnered with Lilly. This is a case where we actually have both a selective inhibitor and a selective degrader being initiated. Our selective inhibitor is in the clinic this year in phase 1 trials, but it's an interesting concept to think about when you would employ an inhibitor and when you would employ a degrader. ARID1B is a very special target for us. It falls into this class of undruggable. That's because it's not an enzyme. The only way to really target it would be to degrade it and remove it. It holds together this BAF complex, so facilitates a lot of protein-protein interaction. But it was really one of these challenges when degradation was set up, is to go after targets like ARID1B. And then our CBP and EP300 programs are great examples of when you can employ degradation to achieve selectivity one over the other to address a variety of oncology indications.
Hop right in?
Yeah.
All right, great. So one of the advantages of having your R&D headquarters located in New Haven is you get to bump into brilliant scientists. And I bumped into years back David Spiegel, and he told me about this concept, and you see some of the important pieces on this slide here. He had this fantasy that he could hijack your liver to do what it does best, and that's remove things from your body and use it for therapeutic purposes. So how do you trick your liver into taking things in your blood, causing disease, and get rid of it? And here you can see a target, and this could be IgG, it could be IgA, it could be whatever target you want. And he created these small bifunctional modes. They're molecular degraders of extracellular proteins. They grab onto whatever you want to remove on one end, and the other end, you'll see this is the surface of the liver, where you have these ASGPR receptors, and there's millions of them. And once something binds to that, it internalizes the target, and you get rid of it. And that's at the heart of this extracellular degrader technology, and it's depicted here in this slide. These bifunctional, bispecific degrader molecules are generally small molecules, but they don't have to be. We can also make the binding end an antibody if we so choose. And any target you can think of in this room that causes an immune-related disease, we can leverage this technology to treat that disease. And what you see in panel one, the first thing, that's an auto-injector, small molecule, so we can get small volumes for patient ease of use. And then in the row two there, you'll see how it works. The bispecific grabs that antibody. Once it hits the ASGPR in the liver, it gets internalized, broken up, and degraded. And now you've removed the target. You can think of the implications as we are identifying more and more autoantibodies and proteins and other things causing disease, that we can remove it. There's advantages of selectivity here, the depth of lowering, and you don't have off-target effects. And so that is a big advantage of this technology versus some of the other ones out there. As far as we know, there's no one else in the clinic with this type of technology. I wanted to give you one example, and there's many. We have a list of 200 potential targets. We have 20 nearer term targets, and we have 3 that are in human testing right now. And the example I wanted to show you is IgA nephropathy. Okay? This is a disease that is caused by excess, a defect in galactose-deficient IgA1. Okay? You get an immune reaction to that, and what you see in this figure is you get deposit and sediment in the kidneys, and then you have an immune reaction that causes all types of problems to the kidney, where ultimately you get kidney failure and could need a new kidney, all resulting from galactose-deficient IgA1. So what if we could go in and just pluck that out and leave everything else there? What impact might we have on the disease? We think it's a potential cure. And to put this in perspective, other companies in this space have all been focused on hitting B cells, where you destroy B cells and you get rid of all the antibodies just to get rid of the one that causes disease, right? Or you have people inhibit complement, where you immunosuppress the patient just to quiet down the immune reaction, in the kidney. We want a surgical approach. And I'm going to show you hot off the presses data of our reductions in humans with what we call BHB 1400. And you'll see that in the light blue here. Within hours, you are seeing robust reductions only in galactose-deficient IgA. One dose, and look, they are sustained for this entire follow-up period here, which is for 10 days, you're getting over, in some maximal cases, over 80% reductions with a 500 milligram dose. And what is remarkable, look at the top end where the 0 is, all the other antibodies are untouched. No effect on IgA, no effect on IgG, IgM, IgE. So this is the delivery of precision immunology. We think we're at the forefront of a new therapeutic platform. IgA nephropathy is one example. If anyone knows anyone with Graves' disease, that's an IgG-driven disease. And in Graves' disease, we chop out your thyroid or give you radioactive isotopes to destroy your thyroid. It's an autoimmune disease. Why aren't we removing the pathogenic antibody, right? And that's what we can do with this technology. Myasthenia gravis, diabetes, early diabetes, you have antibodies to proinsulin that cause your pancreatic beta cells, to die, your islet cells, and that causes type 1 diabetes. So think of all those immune disorders. We want to leverage this technology and bring it to patients. So thank you.
Thank you so much. It's been great to see all this data. It's really exciting progress in the field. So I thought I'd step back and ask Angela, could you just give us a sense of some of the history in this field? Where did this really start, and how has it progressed over the past 10, 15, 20 years?
Yeah, it's incredible to see. Just thinking back to these molecules, the idea of heterobifunctional molecules. So, when you think about drugging proteins, this is a paradigm shift, right, in how you drug proteins. It's a true fundamental paradigm shift. And so classically, inhibitors are designed to inhibit function of a protein. And, when you think about proteins that cause disease, there are a lot of proteins that cause disease that lack an active site. And that's classically what drug discovery was focused on. And so the idea of how do you tackle proteins that you couldn't drug or are under-drugged because they have scaffolding effects and it drives disease. So the real, fundamental change is thinking about what if you could actually bind to proteins within cells, bring those proteins together, right, with a small molecule. This is incredible. These small molecules bring, tens of thousands of kilodalton proteins together. So fundamentally, what if you could bring an E3 ligase to a target protein that you wanted to eliminate and just harness the cell's natural potential to remove toxic intracellular proteins in disease? Fundamentally, that is ultimately a game changer, right? But you could do it by engineering small molecules. This is outrageous, right? So think back to the early 2000s. Sorry, I'm trying to think of when this was. So Ray Duché and Craig Cruz got together, and I think they were, as all good ideas happen, at a pub at a meeting, drawing on a napkin what might be possible. And they came up with this idea that you could recruit an E3 ligase to a target and tag that target with ubiquitin. And then it's just encouraging the cell to do what it normally does, right? You're not inhibiting anything. You're just encouraging the ubiquitin proteasome to do what it does best and eliminate a toxic protein. So you're essentially reprogramming the cell to eliminate a protein. I just think it's fascinating. So the first modality, right, is the PROTAC. It's the proteolysis targeting chimera. This is fundamentally the first. It was published on over 20 years ago. And now look at where we are. So Arvinas, right, is Craig's, startup company, a Yale startup company, funded out of the innovation at Yale. Many of the scientists came from Yale. in the early days. And then, now add in drug hunters, right? Drug hunters are the ones that transform the idea at Arvinas. And some of the drug hunters are right up here. They transformed Craig's idea and Ray's idea into a modality that translated in the clinic and is being presented on tomorrow for the pivotal study for what we hope, is the first molecule to get an FDA approval. So we are really pushing that forward with our partner, Pfizer, for estrogen-dependent breast cancer. And so it's an exciting opportunity. We've put 5 of these degraders into the clinic. And in my entire history of drug discovery, I've been at this for 30 years, I've never seen that many molecules actually go into the clinic and do exactly what we thought they would do. So 3 of these molecules have benefited patients. So we have our estrogen receptor degrader, vepdegestrant, which is in, I mentioned tomorrow you'll see the pivotal data at ASCO. And then we have the AR degrader, ARV-110, and 766, which now brings in a very important mutation in second-line prostate cancer. So very important molecule. So that is now licensed to Novartis. They're taking it into frontline studies and combining it with a very interesting radiotherapy. So I think that's really exciting. And then we've also put in ARV-393, which targets an underdrugged protein called BCL6. It's a rapidly turned over transcriptional regulator. And so it's very exciting that that's going into B-cell and T-cell lymphomas. So it's a very exciting time. And then most recently, we've put into the clinic our G12D degrader of KRAS, and we have a pan-KRAS behind that. So it's a very exciting time at Arvinas. And then we have 2 undisclosed programs where we hope to be in the clinic with those PROTAC degraders next year. But it spawned a mini industry around proximity. Importantly, there have to be 50 companies that started around this idea of proximity, which now encompasses multiple mechanisms, including deubiquitination, RNA splicing, so mechanisms that are so exciting moving forward. So very exciting time.
Yeah, that's amazing. Danette, can you tell us more about some of the different modalities and ways people have translated this basic idea into multiple different kinds of degraders or induced proximity mechanisms?
Yeah, I was joking a few years ago we should start a TAC dictionary.
Yes,
or TACtionary. It's getting out of control. There are so many different variations of these targeting chimeras. In preparation for this panel, I went back through some of the early papers, and actually in 2010, Craig wrote this review in Chemistry and Biology. And with a very simple concept, he said, the molecules of my dreams, right? This was in the title. And he had this pathway of bifunctionals. You could split, you could go to the ubiquitin proteasome pathway, so E3 recruitment or something of the sort, or you could go through the lysosomal pathway. This, of course, was intracellular envisioned degraders, but your pathway feeds into these pathways. And what we've seen over the past several years is just an explosion on what does a degrader look like? Those of us that are developing therapeutics, oftentimes we go directly to the small molecule. We can go to glue development or somewhere in between, like monovalent it's called, or linkerless PROTACs, or heterobifunctional, like traditional PROTACs. There's an entire spectrum there, but now we have antibodies. Now we have oligos or oligo conjugated to small molecule recruiters, or degrading RNA through proximity. Many ideas have come out of this initial concept, and now you see that branching, as Angela mentioned, where the outcome is no longer degradation. The outcome can be whatever biology you want to have to control your disease. But it's really exciting to see the creativity in the field, the embrace of this by not only academia, but industry, both biotech and pharma. I will say pharma was a little, in the early days, they were sitting watching like the biotechs to see how we would do with these monster molecules that we were proposing. But it's really an exciting time. And I think we're just at the tip of the iceberg, even for degraders.
Mm-hmm. Oh,
Yeah.
Totally.
For the extracellular, this seems like a totally different sort of type of thing. I mean, to some degree, you're sort of working with things in a totally different space. So how has that been achieved, getting these to work as well as the intracellular degraders?
Well, thankfully, I'm not as smart as Angela is or Danette, and so that's why it shows the extracellular. There's less problems you can have. Like inside cells, you can run into all types of issues, and you got to be on top of a lot of different things within the cell. Outside the cell, it's a lot easier, right? So, but I'll say one of our big challenges earlier was for investors to understand we were different, right? There was almost this well, why should we invest in you when there's all these PROTACs out there, so many companies in this space, you're coming so late, and you'd be like, no, this is a different area, let me explain it to you. There we're able to, I think, very quickly leverage a lot of the work that has been done in the PROTAC space, right? And use it in some ways a simpler way, extracellular, because you don't have the off-target effects that you have with perhaps some of the intracellular, where you run into other cellular machinery. Extracellular is about the binding region, you bind what you think you're binding to, and only that, right? And then once you understand the ASGPR side, it becomes a modular technology where you can just change out one side, the binding side. And so our biggest focus is on where do we know and have confidence that a specific autoantibody or any target is connected to that disease. And if it's connected, how do we quickly understand what region to bind to so it's highly selective and doesn't touch anything else? So when you look at that galactose deficient IgA data, we want to just pluck out the one autoantibody causing disease and don't bind anything else. And so that's where the scale and focus, I think, is different than some of the other approaches, and that's where our focus has been at. We've been at this for several years, and of course, I'm making it sound simpler than it is. There's a lot of complexities to how do you deliver these bispecifics. If you don't create them with the right affinity on both sides, they either don't bind the target or they don't have the effect at the liver. Or if you have design them the wrong way, you can have effects on the liver, right? So there is complexity, of course, to it. But to Angela's point, this is why you need seasoned drug developers who can help translate this great science. And so again, being here in New Haven, where you can benefit from the science and the academic approach, and then bring a team of people that understand in a disciplined way how do you make molecules that are not toxic, they're selective, and they have the therapeutic effect. Yeah.
You make it sound so simple. I'm sure there's a lot of biological complexity that goes into even the extracellular side of things.
That's right.
So when thinking about protein degradation as a first choice of how to work with things, Adam, how would you go about this is a project that would be good for this, or maybe I should think about inhibition or some other mechanism?
Yeah, that's a hard question. Angela started to address this, I think, in her early response, but it really is all about biology. Mm-hmm. So I think there are cases where there's good data where if you inhibit a target, you have a small molecule that can fit into a functional site. You have a line of sight to develop, something you could get into the clinic fast, and it's going to differentiate. That's the way you want to go. However, If you have a molecule that binds to your target but doesn't have the function that you want, degrading it is a great path forward. If you have a target that not only has a catalytic function, but there's scaffolding function associated with the protein, and you know that could give you a therapeutic advantage, you should try to degrade it. There are tumor cell lines that are treated with, I'm thinking of AR and enzalutamide. The resistance pathway is AR gets up, regulated, AR antagonists start to signal as agonists, right? So the inhibitors become completely ineffective. If you still use that ligand to degrade the target, you're essentially, right, you're negating the resistance that is coming up. And so those are just a couple of examples. I'm sure my co-panelists can come up with more, but sort of Danette's, when she was talking about the SMARCA2 inhibitor versus PROTAC, I'd be very curious to hear your thoughts about what you think about in terms of sort of what approach, maybe not specifically for that program, but how you think about it in terms of whether you want to inhibit or degrade. Because we've been, in our collaboration that we've had with Arvinas, we've been fooled, right? We've thought that there are targets that look like there should be, differential biology on degrading the target, and then you actually make the tools, you start to go in vivo, and the efficacy is not there. And then there are cases where you actually develop PROTAC, and it's 10 times more potent than any inhibitor than you can come up with. So it's biology, what you know, or maybe what you don't know.
Yeah.
Right.
Yeah. Maybe this is a question for anyone. How good are we at this point at designing small molecules or even big molecules that can do this seamlessly? It kind of looks like Legos putting things together, but are we actually there yet, or is that where all the hard work is?
I can comment on, we've been at this for, over 13 years, I think. And all the learnings that go into heterobifunctional molecules, imagine a world where you study those learnings and you incorporate those learnings into your discovery engine, and it feeds the next iteration of optimization, right? So as we, crossed the gastric barrier for oral administration, we have a very large database of PK data. So we've really used and leveraged the PK data in every iteration to design it. I would say for targets, and we've become really good once we have a really good ligand to really optimize PROTACs. And we can engineer in selectivity. We bring in proteomics up front. We de-risk, we get around some of the neo-substrates that can happen with E3 ligases right away. And now we incorporate those machine learnings into our designs of how do we think about that E3 binder. The linkers and how to configure, and now the new data is saying that you don't need a linker. It depends on the target. It's going to depend on the target, of course, and it's going to depend on the E3 ligase and the choice of E3 ligase, right? We now have E3 ligases that confer unique biology where you can now degrade a protein that oligomerizes with the right E3 ligase. So it really is such an exciting time.
Yeah, it's exciting once you know the biology.
Yeah, and once you understand the rules of design.
Right.
Yeah.
Maybe to add to that, on the extracellular side, we've had the similar experience that Angela just described. Spiegel Lab was in it for about 8 years, and we've been in it about 6 years, and now collectively we're at the point where we understand it and are efficient. That, to give you an example, we were able to go from idea to a new degrader in about a year, which is unheard of, right, in drug development, where typically it can take 5 to 8 years to get, to that point. So I'll give you an example. There is a autoantibody disease that occurs in the late term of pregnancy, where you develop autoantibodies to a beta-adrenergic receptor, and it causes heart failure in women. It's horrible. Like these women who are delivering their babies, they think they're going to go home, and they're in heart failure and a very small percent will need a heart transplant because of the autoimmune reaction that occurs. And we know pregnancy is a complex immune, relationship with the child. And so when you deliver, in some people, they get this rare autoimmune disease that looks like it's driven by an autoantibody to the beta-1AR receptor. And we had the idea after learning about this disease and tying it to that autoantibody, and our team put together a degrader in 1 year, and that's one of our assets that we're in the clinic, and we're about to start a trial in. So this is a really transformative time in the field where we can take all of our learnings and now leverage it against multiple autoimmune-related disorders.
Yeah, that's really exciting. I hadn't heard about the autoimmune targets. This seems like an ideal situation where you would want to degrade something in the body that's sort of out of whack. And yeah, that's really exciting. Are there other areas?
I just have one more comment around this too, in terms of what we were discussing about whether to degrade or inhibit. So if you have a ligand for the protein, right, we're actually at the point, Keith, who's right there, could do this without any problem. You could make a PROTAC tool that would probably work in cells pretty fast. It takes a long time and is really hard to make a good in vivo tool. But in order to do the proof of concept test in a cell, you can get there pretty fast to actually understand, inhibition versus degrading. And is there an advantage? Is the biology the same? Is it not? Keith is hiding. Yeah.
He's our head of medicinal chemistry. It's where the magic happens.
I can maybe, I'll also add some commentary here. In the early days, you were excited to see 50, 60% degradation and move from micromolar to nanomolar. Now the drugs are picomolar degraders, right? They're so fast, and they can take down targets for a really durable amount of time. At Foghorn, we work a lot with VHL, and VHL is not represented in the clinic. It's a little bit unmanageable given the hydrogen bond donors. It's very, very difficult to make as an oral drug, so the delivery becomes a challenge. We've worked a lot in our programs to move this degrader to a long-acting formulation, and are seeing really remarkable durable degradation and durable PK with this. But I think, moving away, say, from cereblon and to other E3 ligases and other types of degraders, I think it is the next step, because sometimes cereblon doesn't get you all the properties that you want in a degrader, from selectivity or speed or your DC50. So having more options, I think, in this space would be is what will come next and is needed actually to cover more targets.
All right, awesome. Well, I want to make sure we have a little bit of time for audience questions, so I'll just ask a final question. What kind of investments are you seeing in this area in terms of new platforms, projects? What are you seeing that's exciting? Where are you excited for the field to go at this moment? So,
I could just add to, we're very excited about the work that we're doing targeting, specific targets that are involved in neurodegeneration that aggregate. And so, we are very interested and excited about the advances with different E3 ligases where we can employ those E3s to potentially differentially degrade proteins that were not something you could imagine degrading, like tau, alpha-synuclein, mutant Huntington. So game changers around neurodegeneration where the biomarker paths are really clear. I think that's an exciting opportunity. We're also targeting other proteins. We talked about scaffolding function in cancer. And, we think they're very important drivers of disease. And so that's a huge opportunity. New ideas around how to do what Vlad's group is doing from an intracellular perspective and engage the ERAD system, I think that's an exciting opportunity as well, because if you could get certain proteins as they're being secreted out, you could potentially target different proteins differentially. So there's a lot of exciting stuff going on in RNA regulation as well. So, yeah.
Okay. Lots to look forward to.
Yeah.
Vlad?
Yeah, I think we benefited from having a little bit of an advantage of there weren't a lot of other extracellular degrader companies, but what we're seeing now, because the potential of these, as you can tell, is pretty immense, and most of these represent multi-billion-dollar markets, we are seeing increased competition now within the extracellular space. Several new companies successfully funding, I think one last week, raised over $100 million. So we expect we're going to see more and more of that. Which I think is healthy and good for the space. I'm also happy that we've had a little bit of a lead because it is a tricky space as much as I joked about it being simpler. We've learned a lot about how to optimize these, and we're already moved on to our third generation kind of optimized versions of it. So I think the race in extracellular is going to all be about selectivity, depth of lowering, small volume sizes so they're very convenient for patients, and a high degree of safety, right? And if you can do that, you're going to have a really successful platform.
Awesome. Danette?
Yeah, I'm really excited about continuing to apply degradation to oncology targets that we listed. Those are all preclinical programs, so we'd like to shepherd them into the clinic. But also, now that we've learned how to work really well with bifunctional molecules, what else can we do? So expanding in the induced proximity space, especially, we work with a lot of chromatin regulators. There's been some exciting work out from one of our co-founders, Jerry Crabtree, and Nathaniel Gray's group about rewiring transcription by moving around chromatin modulators. And we actually did a pilot study that we submitted on bioRxiv to move our BAF remodeling complex to a new location on DNA to activate genes important for regulation of sickle cell. So there's a lot of opportunity, I think, in what we have learned from degraders to start in these other areas of induced proximity.
Adam? Yeah, and maybe just to follow up on some of the things that Danette has said about induced proximity. Craig and Stuart Schreiber have really been the leaders in this field. They will win the Nobel Prize. There's no doubt. It's just each October, it is the safe bet when it comes around. But proteins, when you actually can get a small molecule between them, tend to stick together in a very potent ternary complex. And that is incredibly impactful and extensible for doing all kinds of things. You can do it where you choose to degrade the protein if you make the ternary complex between a protein and an E3 ligase. Therapeutics, which is an additional company that's come out of Craig's lab, is using this to target overexpressed AR in prostate, linked up to an effector protein in a very tight ternary complex. Their lead program is just in the clinic this year. That looks like a really good compound from what I've seen. They're transcription factors that we don't know how to ligand, but you can take sort of a large presenter protein and modulate the surface with a small molecule and actually have it inhibit the protein which you don't know how to ligand. There are companies that are doing this in both the intracellular and extracellular space. And there are 5 more things that I'm thinking of that I can talk to when I'm done. It is the most powerful therapeutic concept, I think, that people have, this is coming out of, I think Craig and Stuart are really the leaders in the field, but we are right on the cusp of driving new modalities for therapeutic targets that we know are validated that we haven't been able to access yet.
Fantastic. Thanks so much. So I think we have time for a few questions in the audience. I see someone down here. Can you help?
Speak up really loudly if you want.
We have microphones, I think, but...
If not, it's okay. So I'm Noelle. I'm a postdoc here at Yale. And one of the questions I have, because I think this is something newer than what we normally see for, in this space of breaking down protein, what are the adverse effects that you're seeing? Because something that I see, especially with the extracellular one, where you're utilizing the liver. Is there limitations to these mechanisms, and do you see any adverse effects that are different that we normally see with other small molecules?
I'm happy to jump in on that part of it. So, it's interesting. The big concern from investors was always that you would have some effects, on the liver. We also presented yesterday, didn't have in the slide deck, our EDISH plots looking at, the liver toxicity outcome measures, and it was all clean. So I think when you develop the molecule the right way, there are not effects on the liver, and your liver does this every day, right? But just to be clear, you can develop a molecule that's hepatotoxic, so you have to that disciplined drug development approach of knowing what to stay away from and how to design them the right way. But if you do that, we've seen nice translatability from the animal models where there's a lack of toxicity in the liver, and the human data that we have to date. Now, if you bring certain targets to the liver and you overwhelm it, like perhaps maybe cholesterol, like let's say if you want to remove cholesterol, probably not a good idea. You probably could create fatty liver, right? By doing that. So I think you have to know what the target is, know how to design the bispecific, and so to keep that in mind to avoid those toxicities. Outside of that, I have to say it's been exceedingly safe and selective and targeted. So we haven't seen other things. The major thing is it's a needle going into a patient, so you get a little bit of a injection site reaction that's you get with all injectables, with the GLPs and everything. Oh, maybe just on immunogenicity. So it's interesting. This mechanism is probably tolerogenic, and so we haven't seen yet, immune reactions that you typically might be concerned about with this type of mechanism. But it's something you'll have to think about as you increase dose load of these things.
Great, thank you. Any other questions from the audience? Oh, up here?
The producer.
Just getting our steps in.
Sorry about that. This is another question for Biohaven. I'm wondering, have you seen the work on catalytic extracellular degraders, and where do you see that space moving?
Yeah, no, yes, we have. And I think that has to do with, the duration of effect that we talked about earlier. So I think there's advantages to catalytic degraders that allow these molecules to be recycled and have their action again, so you don't need to re-administer them. For some targets, that will be important. For others, it will be less important, because once you remove the target, it's the regeneration rate of the target that determines re-administration. So for example, with IgG, you're looking at normally it takes 21 to 28 days for you to regenerate IgG. And so there you would not need a catalytic degrader, because you're able to hit the target, bring it down, and then re-administer it intermittently just to keep it down and maintain. So I think there are a lot of considerations when you choose your target, whether you need a catalytic approach or not.
Hi, my question is for Adam and anyone else who can answer. Can you elaborate a little more on the limitations of targeting transcription factors with this technology?
Yeah, so transcription factors, at least ones that are very well validated, tend to have a lot of intrinsically disordered regions of the protein. And at least a number of them seem to be, succumbing to Dell screen, where people are starting to find ligands. Chimera just made an announcement where they have an IRF5 PROTAC, and I'm sure people have tried the ligand to IRF5 for a long time, and I'm sure they did it in a Dell screen, so that's actually quite amazing. But you can't do that with most transcription factors. So to use this sort of templated protein in order to bind very tightly to a transcription factor to inhibit its function or to traffic it away from where it's causing sort of the effects in the cells that you want to prevent is something. It's actually the original Stuart Schreiber concept around, his FK506 work that he was doing in the 90s and stuff. And I think only now people are appreciating that this could be general, or at least more general than people appreciated. So folks are on the lookout to, find these presenter proteins that potentially can make these tight ternary complexes with transcription factors to affect their function, I think.
Yeah, and I was just going to add, that was great, Adam. Just one of the factors, at least for PROTAC targeting of transcription factors, is to overcome that rapid resynthesis rate that transcription factors can be challenging with a PROTAC. We've been able to do it with BCL6, so that rapidly resynthesizes within an hour. And then they can also be highly upregulated by inhibition. So a PROTAC modality turns out to be a great way to target transcription factors.
Yeah,
Thank you.
maybe I can just add to, on Adam's favorite topic, covalent binders.
My second
yes.
favorite topic for this panel.
It's PROTAC.
Proximity.
But this is having some traction on transcription factors. The big challenge there will be selectivity, especially if you're targeting something in these unstructured regions. But transcription factors are natively degraded by their native E3 ligase. So there's a lot of efforts to try to figure out which native E3 ligases are paired with which transcription factors. And then using the paradigm, Schreiber and others, and a paradigm from what we've learned from imide, or cereblon-based glues, is can we just shift that KD of that native interaction to strengthen it to really drive then the degradation. The challenge there really does lie in figuring out what those native relationships are with the E3 ligases.
More questions? We've exhausted everyone, so thank you so much. Everyone, please join me in giving a hand to our panelists. Thank you so much for